Hello. Today what we'd like to do is just cover some of the aspects of the CNCPS platform moving to 6.5 biology from 6.1. There's been some confusion over what is actually changed and how it impacts our recipes and our diets. So today we're having a discussion between myself, David Weber, got Buzz Burhans on the line, and also Ermanno Melli. And what we'd like to do is just talk over some of the changes that we're seeing adopting the new biology. So right now I'd like to go to the next slide and talk about the carbohydrate changes in there. Buzz, do you want to kind of help me go through some of this?
Sure. So there are significant changes in 6.5 to some of the factors related to carbohydrate nutrition. The three major ones are listed here. Number one, the calculation of NDF degradation rates is now based on a different method that uses 3 time points. Previously, in 6.1 and earlier, the degradation rate for carbohydrates, NDF, was calculated using the Van Amberg method, which involved the use of a single time point and a lag. And the lag was specific to each laboratory. Now, the labs are reporting 3 time points, and the time points for forages are 30 hours, 120 hours, and 240 hours. Also, the same system can be used with different time points for concentrates and byproducts. The concentrates and byproducts use time points at 12, 72, and 120 hours. And these calculations are now done internally in the NDS platform so that they generally do not show up on the samples that you receive from the labs. The time point values will show up on those samples. The KD for NDF will not show up on those. If you see a KD for NDF on a sample, it is calculated using the old system, the Van Amberg system, with the single time point. And we'll show you in a minute where the KD is calculated in NDS. The second important change in 6.5 is the undigestible NDF. The undigested NDF is estimated as the residual from the last time point in the series. So for forages, the last time point is 240 hours. So the residual in the assay at 240 hours is considered to be undigestible. For non-forage feeds, that time point would be the 120-hour time point. So for concentrates, for instance, it's at 120 hours. This replaces a system that's been in use since the beginning of the CNCPS model, which estimated the undigestible NDF as the value of lignin times 2.4. This new change to the estimation of the undigestible NDF is an improvement in the digestibility assessment of feeds and has improved the calculation of energy values significantly. And thirdly, the NDF itself is now expressed differently. It's now expressed as ANDFOM. So as previously, NDF is still assayed using amylase to free it of starch and using sodium sulfite to free it of nitrogen and protein. But the new change in 6.5 is that it is expressed as an ash-corrected basis. So it is ashed and it is expressed as ash-free. This means that the value of NDF is going to shrink because we are no longer reporting the ash content which is associated with the NDF portion. For most feeds, this is not a huge difference. The NDF pool will shrink slightly. However, for some forages that have a high amount of soil contamination, this will be a significant difference in the NDF. So we might go over and look at the NDS program and look at where these three items are resident. Dave is going to take us over to NDS, and we'll look at a feed sample perhaps. And when we get there, we'll show you where these things are input. So Dave, if I go to the constants calculation, is probably the easiest place to look at this, and you'll notice here on the ANDF value, the ANDF is now called ANDF-OM, and that value is the ash-free value of NDF. Then the other thing that is on this page is, you'll see if you look down, you'll find the carbohydrate B3. Carbohydrate B3, or carbohydrate C, I'm sorry. The carbohydrate C is expressed twice there. And maybe Dave can put his mouse down there. It's expressed once as UNDF lignin times 2.4. That is the original value that we are used to seeing in earlier versions of the model. And it's also expressed as carbohydrate C UNDF, and that is the new value that is expressed at the 240 time point. So you see in this example that those two values can be significantly different. In this case, for about 4 points different. And that value is used to calculate the potentially digestible NDF. So you see that in this case, when it is determined using the UNDF system as opposed to the lignin times 2.4 system, there is more undigestible NDF using the UNDF system, which means that the pool of potentially digestible NDF is going to be smaller. So that's also going to make a difference in our energy values. The energy values are also going to depend, as we said, on the calculation of the rates. So if we go over to the NDF digestibility page, this is where the difference in the rate calculation enters. And perhaps Dave can take us over to there. Here we see that this particular sample has the three rates from the lab entered. So we have a 30-hour rate, a 120-hour rate, and the 240-hour rate. And in this case, when those three rates are either entered or imported from the lab XML file, the calculation of the KD for the NDF is automatically made using the Raffornato rate calculator. If we were to have a sample that had a single time point, for instance the old 30-hour time point alone that we used to see, if we also knew the lab's lag time and entered that, where on this slide you see the 1.61 value. So if we had entered the whatever lab this came from, that particular lab's lag time, it would calculate the carbohydrate B3, the NDF KD, using the old Van Amberg rate calculator. So that's still present. And in NDS knows whether you have a single time point and a lag or the 3 time point entered, and depending on what you have in there, it will calculate the appropriate KD. We should note that as we move forward, the library values for the NDF-KDs are based on the Van Amberg calculation. And that's because there has not been enough of the new 3 time point work done yet for the databases to have a large enough population to have populated the library values. So it behooves us all as practicing nutritionists to go ahead and order these 3 time point values from the laboratory, and in most cases, we'll cover this again in a later tutorial, but in most cases that means you'll have to be sure to order the correct NIR assay or wet chemistry assay from the lab. In some cases, if you order and you're not aware that they have new offerings which include these 3 time points, you'll get the old system there. So these are the 3 places where these carbohydrate fractions will show up on the feed side. If we go over to the ration itself, there's one other place that might be worth noting, that this UNDF 240, the undigested NDF, is going to change a value. If we look at these UNDFs that Dave is pointing out, the green fields where it says UNDF-I as a % of body weight. So with the UNDF value now calculated at 240 hours for forages, these values are going to change a little bit from the old lignin values. And the reason is obvious. If you remember that sample that we were looking at, for instance, that's only one sample that's in this diet. But the UNDF value was now 12%, and the lignin times 2.4 and UNDF was 8%. So you'll see that we have more UNDF, and when we express that increased amount of UNDF in the ration, it means that our UNDF intake as a % of body weight is going to be increased. So this is something to be aware of when you're evaluating the UNDF as an indicator of the dry matter intake potential.
And so, Buzz, I think when we get to running another tutorial that we can send out on carbohydrates, we'll go and change that forage from the 6-1 using a single time point with that lag versus the new calculator with the 3 time point, and we can show the impact of that in the diet.
I think that's a great idea, Dave, because all of us are going to have to get used to the fact that although we're dealing with the same terms, NDF for instance, or UNDF, the amounts that we have as references or benchmarks for what we're shooting for or what we're expecting are going to change a little bit with these changes in how the assays are done, how the calculations are done. So we'll look forward to doing that in our next tutorial. I think the next one we're going to look more in depth at this carbohydrate issue. But for the minute, we probably should go back to our slides and look at some of the changes that are related to the protein aspects of 6.5. So with 6.5, there are several significant changes that are related to protein. And perhaps the first is that amino acids in the model used to be expressed basically as a percentage of the undegraded protein, the insoluble residue from the assay. And they are now expressed as a % of crude protein itself. And that will make it easier for us to change those values. Not that most of us are doing many amino acid assays, but certainly for some of the suppliers that have feeds in here, they will be able to update this more easily, as we will as well if we do that. A big change is that the amino acid efficiencies are different. Now, efficiencies basically describe how much or what proportion of the amino acid supply ends up being actually utilized for product, whether it's tissue or milk. And because 100% of the absorbed amino acids don't end up in milk and tissue. And historically, the model has applied one efficiency for the maintenance functions and one efficiency for lactation. In 6.5, these two functions are now combined as far as the efficiencies go, and a combined efficiency will be used for the amino acids, whether they are used for maintenance or lactation. It's all one efficiency now. We'll look more closely at this in another tutorial probably in 2 weeks, and we'll see that those efficiencies, when we combine them, are lower, and that has some implications for the requirements. Our requirements are going to be a little bit higher, mainly because the percentage of the amino acid that's absorbed that is utilized is going to be lower because of this change in the efficiencies. That's an important change in protein nutrition in the new model. And the third change here is that the unavailable and undigestible protein, the protein C fraction, can now be determined by the Ross unavailable nitrogen assay in 6.5. This replaces the use of ADIN, and it applies to non-forage feeds only. So, for instance, blood meals and heat-treated soybeans or canola meals, for instance. These are feeds, particularly the blood meals, they don't have fiber in them. There's no acid detergent fiber in blood meals, so it's more appropriate to use this new assay. And work at Cornell in the Van Amberg lab has shown that this assay the use of this assay to determine the undigestible fraction, that protein C fraction, can really make a difference in some cases in the prediction of the available protein to the animal. For forages, because they do contain fiber, the original acid detergent insoluble nitrogen, so the nitrogen that's bound to the ADF fraction remains an estimate of the unavailable nitrogen. And although it's not new to 6.5, we remember that the protein B2 rate in forages is now set to the carbohydrate B3 rate. And that makes sense because most of the protein that is available at a moderate rate in the B2 fraction is resident in the associated with the NDF fraction. So it makes sense that those should degrade at the same rates. Maybe if Dave can take us back over to the model, we can look at a couple places where you'll be able to observe these changes. So if we look at our, I guess we've lost our feed. If we pull up a feed here, we'll be able to see in a minute some of these changes. Okay, when we go into the feed tab, the easiest place to look at these is to go into the constants calculation sub-tab there, and then look at the protein fractions. In this protein fraction here, if we go over maybe to the amino acid section. So here's our list of amino acids, and the amino acids, as we indicated, are now expressed as a percent of crude protein rather than a percent of the insoluble residue. We can't really see, I don't think, anywhere the difference in efficiencies here, but in our tutorial coming up on the changes in protein and its implications for formulation and prediction, we'll tabulate what those changes are for the amino acids, and we will see the difference and the fact that the new efficiency values are generally lower. The other thing, if we go back to the protein tab here, sub-tab, it is possible in that ADIPC field on this on this tab to enter a laboratory value from the RUM&N assay. Now there is some question, and we'll get into this a little bit more when we do the tutorial specifically on the changes in the protein fractions, but there is some question about how ready the labs are to run that RUM&N fraction. We'll talk about that a little bit more. For most of us, this is not an assay that we're going to run very often. It's fairly expensive.
Let me grab a blood meal or something out of there so they can see that. Should have had one of those open.
Okay. The reason Dave is going to do that is we're looking at a forage there, and that RUM&N assay really relates primarily to the concentrates and not to the forages. As we said, the forage unavailable protein or unavailable nitrogen remains calculated as the association with ADIN, or the acid detergent nitrogen. We'll get Dave to open up a blood meal here, and here we can see that the screen basically looks the same, but it would make more sense for us to be changing this if we had an assay value in that ADIPC value there. You see that there is a check mark there for the RUM&N, and that can be checked, and you can enter your value. And notice that when you check that value on the blood meal, the label for that field, the third field down in the list, is now labeled UN RUM&N. We could enter a value in that. And let's uncheck that just so everybody can see that. It's highlighted now. Dave's got his cursor on it. When we uncheck it and then go back and highlight it, it should go back. Maybe you can re-enter the, yeah, there we go. So if Dave, you highlight the ADIPC line right below that, where we have highlighted right now. So that ADIPC line, you notice that that label has changed. We got on my screen the NDIP is highlighted, but the field that's right below that, yeah, there we go. Notice that when the UN ROS checkbox is unchecked, then this defaults to the label of ADIPC, or the acid detergent insoluble protein fraction. When we check it, it goes to the ROS fraction. But it is enterable either way, and is a difference. I do want to point out one other thing that's on this. So in this, there's another checkbox at the bottom of this table, and that's the KD for the protein B2. And Ermanno pointed out earlier today that checkbox is not appropriate to be checked for all feeds. And maybe, Ermanno, you could explain the difference there, because this is a good example, having this particular feed up here. Ermanno, are you with us? Could you just comment on when that KD for the protein B2 should be set equal to the carbohydrate B3?
Yes. This should be checked. This checkbox should be checked for forages because the NDF content is high, and as you described, Buzz, this fraction, the protein B2 fraction, is strongly related to the carbohydrate free. So in this case, the suggestion is to check this checkbox. For other kind of feeds, for non-forage feeds, for concentrates and byproduct, mainly the approach is similar, but not for all feeds. So our suggestion is to check this checkbox only when we are working with some non-forage feeds with a significant amount of NDF. For example, in this case, you are working with a blood meal, and it is irrelevant to check this checkbox because you checked the UN, and in this case, the protein B2 are 0, so it doesn't matter to check this checkbox.
Great. Thanks for that explanation, Ermanno. Dave, maybe you could take us back to the slides, and we'll just touch on the changes in the feed library, and then sum it up with a couple of comments on the NDS implementation. The most recent feed library that has been released over the last year involves significant changes to the size of and the amount of different fractions in the feeds. This is to a certain extent independent of 6.5. That library was updated beginning several years ago. But what is particular to the changes in the new library that relate to CNCPS version 6.5 is that the amino acid content is now expressed in that library as a % of crude protein. In the old Cornell base library, it was expressed as a % of insoluble residue. So that's the big change, and that makes it consistent with how the calculations of amino acid supply are now made. So that's the key changes that have occurred in 6.5 compared to 6.1. We are going to go through the implications of those changes for formulation and predictions in 2 additional tutorials in the next 2 weeks. One will be on the carbohydrate implications, and one will be on the protein implications. But rest assured that NDS Professional is running the current and most updated CNCPS 6.5 version. It is the complete and exact implementation of the underlying biology and equation set as released by the Cornell Modeling Group. And that's important to understand. It is complete, it is implemented, and NDS it is the leader in that implementation. The implementation, the model itself, is complete and final. And NDS was the first, and is the first, that has got this new version of the model out into the field in a commercial platform. So we're very proud of that. And part of the reason that has happened, part of the reason that NDS is leading the field are some of the things here. First of all, NDS is developed and maintained by practicing dairy nutritionists. Ermanno and his crew and Dave and I, we're all out in the field actually working with dairies, and we know how valuable it is to have the best and most current science available. It makes a difference, and Dave was commenting just a little while ago about it's made a big difference in some of the herds that he's worked with, and I would support that. It's true. The other thing that you might be interested in knowing is that NDS has a fairly large programming staff. There are multiple programmers on our staff, and indeed, as far as we're aware, the NDS system is supported by a larger group of programmers than any of the other commercial softwares out there. And these guys are great. And those of you who, some of our users have had an opportunity to interact with those guys, they are just outstanding programmers. They're very talented. and there's more than one, so we're quite proud of them. And then our tech support staff, and again, that's going back to in NDS North America, Dave and myself, and with access to Ermanno and to the folks at RUM&N, to Elena and the programmers, we're all involved in the field. So we think it makes a difference, and we're proud of being the leader in the implementation of the new 6.5 biology. And we thank you and appreciate your endorsement of that leadership in your use of NDS. Dave, you want to comment on these tutorials coming up?
Yeah, I think our goal for today was just to review and take a little of the information that's in the field to show you the guidelines of what's changed, where it's changed. The idea is that what we plan to do is redo some tutorials, some in-depth tutorials, to present the information on how the carbohydrate, step-by-step, these interactions that have changed from 6.1 to 6.5, where they are in the model. One thing that Buzz was mentioning was the programmers working with the new biology. It's in the program. It is available there for you to use. We'd just like to clarify some of the confusion. Doing a tutorial on carbohydrates first as one single tutorial, release that, and then also go back and look at the amino acid implementation in 6.5. So those will be coming up. And the third tutorial that we'd like to release is just reviewing some diets, some back and forth, looking at the rations, looking at how those changes might impact the diet. So we appreciate your time. Thank you for viewing this. And again, we'll be back with more information in the coming weeks.
Thanks, Dave.
Thank you, guys.
Thank you, Dave.